Okay, so welcome, folks, to our monthly CASP special interest group on ensembles of conformations. Um, so I think a lot of people, a lot of people have been coming regularly, but some people may be new. So I'm just going to give a brief introduction to what we're trying to do. Um, CASP has traditionally been an approach to validate methods of modeling and structure prediction, and it's had a tremendous impact on science and the community, particularly as seen in the last few years with AI-based methods for protein structure modeling, which really were enabled to a large degree by the ability to have blind data to test against and validate against that CASP provided and an infrastructure for that. So CASP has spun out a couple of special interest groups to focus on sort of the next challenges beyond generating a static structure. And one of these is this special interest group on ensembles of conformations. So we have an organizing committee and we put together a vision statement recognizing that biomolecules don't have static structures, but rather adopt a distribution of conformations. This CASP SIG aims to evaluate if it's possible to do a critical assessment of prediction of conformational ensembles in multiple states. It really is challenging to understand what is the ground truth and how would we assess such modeling? And not only the question of how do you do such modeling. So we will look at developments in the field that are anticipated in the next years and define protocols for assessing the accuracy of multiple or ensemble model predictions. And it's very much an experiment to do this. So we put together a steering committee, Egzi Taraka in Turkey, who's led some of the complex prediction assessments, Damiano and Silvio, who have been involved very much in intrinsic disorder and protein conformational ensembles. Emily Tai at NIH has long been involved with CASP and assessment. Wim Rankin, also very much an expert in intrinsically disordered proteins and flexibility. And Shoshona Wodak, who's one of really the leaders in thinking about this area of multiple conformational states and has been involved with CASP for quite a few years. So we have this expertise and help, and of course the CASP organizers, John Moll, Christoph Fidelis, and Andrei Christofovich. And if there are other people who want to get involved in organizing, we are open to adding more people to this group. So when we talk about multiple conformational states of the proteins, this could be anything from like a two-state model, like traditionally one describes the two conformational states of enzymes like adenylate kinase, to a wide distribution of states as one thinks about intrinsically disordered proteins or linkers. And in fact, we're going to hear today about some ideas about assessing multiple conformational states of linkers between domains, which is kind of a hybrid between intrinsically disordered states and ordered states. So it's actually a class kind of its own and special in terms of the kind of data that you can get to characterize such states. And just point out that this special interest group is one of three that CASP has organized. There's also a special interest group on AI methods in structural biology more generally that's organized by Arnie Ellefson, and it's on the second Wednesday of each month. The next meeting next week will be Sergei Achinnikov and Patrick Bryan on methods of using AlphaFold. And then we also have a CASP special interest group on RNA, modeling of RNA, which even includes some multiple conformational states of RNA. Rachel Kresh and others organized that. And that's, I think, is the second Tuesday of each month. There's actually a schedule online here. And I believe it's now set that the CASP meeting for 2014, which I guess is CASP 16, will be November 29th to December 3rd. The location hasn't been set yet. So just so people are aware of that. So today we have presentation on this general challenge of dealing with interdomain linkers. And Bruce Donald and Terry Ois from Duke University have been exploring this and actually have published some really groundbreaking results in this area. So they're going to tell us about the structure of a two-domain protein with a flexible linker, a continuous distribution of interdomain orientations derived from NMR residual dipolar couplings. And after the presentation, we'll continue to discuss for 15 or 20 minutes how we can evolve this into a CASP type challenge. So with that, I'll let Terry and Bruce take over, and you guys can present your team and describe your ideas. Go ahead.
Okay. Can you, yeah. This will stop. So you're still sharing, I think. I don't know.
Go ahead.
Let me bring up our title slide here. Okay. And I think before we start, I'd like to introduce the three of us who are here related to this project. One is Bruce Donald. My video isn't actually, there we are. Okay, so Bruce Donald is my collaborator in this whole project, and we'll be presenting much of the deep theory behind this project in just a few minutes. And Elaine Mupwali, sorry, Elaine, is also on the team. Hopefully you can see her, at least her picture. And she's the student who is planning to collect the new version of the protein we're talking about today. So that's our team at the moment. And I will then proceed to introduce our system. I guess I missed the title slide, but Guy just gave the title. So on to discussing the protein of interest that got us thinking in this direction. I've been working on this protein for over 20 years at this point, starting out working on it as a biophysical model, picking up actually on a series of experiments by Guy, and doing protein folding experiments with the B domain of protein A. This is the sort of roadkill version of the protein, stretched out. It's a 524 residue protein that is highly abundant on the surface of Staph aureus and is a multi-virulence factor. It binds to lots of different proteins, not just antibodies, which is what it's famous for. It also binds to 4 or 5 other host factors. And for that reason, and given the importance of Staph aureus in understanding bacterial resistance, antibiotic resistance, we've been focusing on this protein for quite a while. Back in the mid-2000s, I started to think about what the B domain does in the context of the full-length protein. You can see the domains here. There are 5 of them. They're nearly identical in both sequence and structure and function. And they all bind to the same factors. They all are 3 helix bundles, very similar in structure. 4 of the 5 domains have had their structures determined by one method or another. They have very conserved sequence. The bold letters in this slide are residues conserved with the central domain, which we think was the original domain that was then gene duplicated over the course of evolution to make 5 of these things in a row. And so you can see how similar the sequences are. The other thing that's common to all of the domains is the linker between them. It's almost identical in sequence, except for this one that has this AQQ insert. And that linker has been pretty much overlooked prior to our work on the multi-domain version of this protein. Our first hint that there was something special about this linker came from our thermodynamics studies, which showed that two B domains linked by this conserved linker, KADNKF, that linker managed to completely decouple the two domains from each other thermodynamically. In other words, they both fold and unfold with exactly the same kinetics and the same thermodynamics, whether they are connected together or whether they are separate. And that suggested that there was something special about the linker. And then we further did relaxation experiments with a 5B version. So it was a mimic of what we call SPA-N, the 5 domains of the N-terminal half of the protein. We made a 5B version and determined the relaxation parameters for all of the residues in that 5B construct. We used 5Bs because it simplified the assignments. And what we learned is that the domains themselves are highly ordered with order parameters above 0.95. But the linker between them was very disordered, that is, very flexible. I had very low order parameters, well below 0.7, okay? And it was on that basis that we realized that what we were looking at was a protein that had 5 well-ordered domains that had flexible linkers in between them. And that, of course, immediately begs the question, well, what's the structure of this protein? We couldn't answer that question with conventional methods because the very flexibility leads to a very wide distribution of conformations. But it was still relevant to ask, are those distributions of conformations completely random, or are there preferred interdomain distances and orientations? The linker is quite short, but the orientation is not a priori known. Our first experiments to answer that question were actually Sachs experiments carried out at APS on beamline 18. And what we learned from fitting the data of a series of constructs ranging from 3 Bs in a row to 5 Bs in a row and SPA-N, what we learned from that is that we could fit those Sachs profiles to a polymer physics model that's called excluded volume pearl necklace model. And that model allowed us to describe the persistence length, that is the stiffness, the Flory coefficient, which is sort of a measure of the compactness in aqueous solvent, and the radius of gyration of each of the individual domains, which were represented in the model as pearls on a flexible pearl necklace. And then that led us to take that philosophy of fitting data to a minimally parameterized model to then investigate the use of an NMR observable called residual dipolar coupling. And for that, I'm going to turn it over to Bruce, and he's going to give you some background on RDCs and what they can tell us, and the basis of the theory of the model that we use to interpret that data. So I'll stop sharing and let Bruce take over.
Thank you, Terry. I'll share my screen here. Okay. So it looks like that's working. It's a pleasure to be here with you all. Just wanted to give a little manifesto for continuous distributions and why they might make sense here, as Terry introduced. Here we have interleukin 14. Even looking at motions in the rotameric wells, you see a wide range of energies ranging from good to bad using Richardson probe dots here. So proteins, all biophysics tells us they're actually continuous entities. And some model of continuous distribution probably makes sense here. From a modeling point of view, of course, we have ways of thinking about ensembles here, some from MD, from a recent paper, even looking at really detailed snapshots. How can this be used for ensemble data? Let's look at what you might call the traditional approach. This is a very broad cartoon, which is you make a big ensemble by Monte Carlo or molecular dynamics. And then given this thing, you want to back calculate observables by averaging, because all you can observe is an average in an experiment. And of course, the physics of the world is computing a real-time ensemble average, and you compare the averages. They won't agree, so fitting comprises deleting members of the ensemble typically and then iterating. So this model has many, many degrees of freedom, the degrees of freedom of the size of the ensemble and all the degrees of freedom of the protein here. So there are issues with this approach, right? One is the discrete ensemble isn't continuous, leading to the rather alarming observation that the density in terms of probability is undefined between discrete ensemble samples, or maybe it's zero. We propose instead, let's model a continuous distribution, get rid of the discrete samples, which we don't need. And of course, we can sample that as much as we want to have a particular conformation. Other issues, and these were brought up very nicely in Susan's talk, has to do with non-uniqueness. So let's look at a toy example. Suppose we have a two-dimensional configuration space and a linear transfer function. If we have only one protein structure, it has some data we can record from that. But of course, if it's average data, and this is an ensemble of two structures that gives exactly the same value. Here's another ensemble. In fact, there are an uncountable number of ensembles that give the same data, even in this simple case here. And this leads to all sorts of problems in the model having many, many degrees of freedom. You can also hallucinate things. For example, if an adversary were to say that all these points here were the actual set of the ensemble, nature could collaborate by just adding this in. This also averages to the same point. So with average data, it's highly problematic if your model has many more degrees of freedom, easy to fit. It's possible to overfit these. And if you would like examples that I don't have time to present from the literature of models that have hundreds of thousands of degrees of freedom, even though we only measured 80 things, we can easily look at these. So this is statistically very problematic. So basically, I want to illustrate it with this cartoon. Here's our dinosaur as a cloud of points and means and all sorts of statistics. All these clouds of points have exactly the same to 2 decimal point means and medians. And this is the situation we have in trying to edit MD or Monte Carlo ensembles, is that we're at an average. And so we want to, nice thing about minimal parameterization, so we can have low dimensional models that have roughly a low enough degree of freedom that we can fit the data confidently. Okay, so our idea is to have continuous distributions based on the data. So basically the first thing you do in any science experiment, in normal science, you make measurements and fit a Gaussian to them. Data from NMR, the Gaussian's over orientation, so rotation space, which I call SO3, and we can fit a Gaussian or mixture that is sums of Gaussians, and we can use that for all sorts of things like calculating thermodynamics, as Terry said. But for now, I'm just going to talk about rough outline of fitting a single Gaussian with a single mode just to see how this might work. Okay, so let's look at our setup here. Is we have 2 domains, as Terry said, relatively rigid compared to the linkers. We engineered a lanthanide binding tag here, so that's a protein design problem, and we carefully verified that it doesn't perturb the structure using both HSQC data and other observables. And we want to know the relative orientations of these under broad averaging. The lanthanide is used to align this in the magnetic field. I'll talk about that in a minute. So again, our cartoon here is we have 2 domains connected by a linker. The difference in the pose is, of course, a translation and a rotation. We're only interested, well, we're interested in both, and we can sort of deal with that, but we're only talking about rotations in this talk. So what we're trying to develop as our model is a probability density function over rotation space, which I'll denote by SO3 here. For those in NMR, it's the same as SU2, basically. So basically, the orthogonal matrices that do rotations. So that's the model we want to get here. So this is the only physics slide we need, but the rest is covered in my textbook. So imagine we do these residual dipolar coupling experiments. They actually measure in the molecular frame both the internal motions of vectors and the global tumbling or orientation of the molecule. And these are combined by 2, if you will, alignment tensors here by a kind of multiplication called Frobenius multiplication, because we have relaxation experiments that say that we can treat this, the T is based internal motions as being negligible compared to the inner domain motions. We're really only interested in this one S, which is the overall orientation here. So this is the observable we fit to this, and I'll say more about that in a minute. But this is the basic physics of RDCs that we need to know for this. Again, covered in chapter 5 and others of my textbook if you're interested in learning more, and in our paper. So this is an ill-posed problem in the sense of Hilbert. That is, we are trying to, physics of the world is calculating the RDCs, which are these values here, from the inner domain orientation distribution. We want to invert that somehow. We can't invert it exactly, so we add assumptions to regularize that. And I'll talk about what those are. Hint, Gaussian. Okay, so here's our situation: is the engineered lanthanide binding tag binds a lanthanide, actually various different ones. This more or less restrains the red domain, and its alignment tensor that measures its averaged orientation is S1. Then the second domain is free more or less to rotate within the constraints of its tether. We're interested in the relative motions of the red one and the blue one, having decoupled it by this lanthanide paramagnetic tag in the magnetic field. So what do we have here? What are these alignment tensors? These are real and symmetric, and they're traceless, so we can decompose them like this into a rotation, a diagonal matrix of eigenvalues. So all the alignment tensors have this sort of structure here. Again, U is just a rotation. I'll talk about representations of rotations in a moment. So again, for now, you can think of a rotation as being a 3x3 matrix that has orthonormal rows and columns and determinant 1. We'll get better things in a minute, but we have basically, at a first approximation, all these degrees of freedoms in a rotation modulo the orthonormality. So the rotation describes the relative orientation of the domains. And just a little review of what we'll be doing. Obviously, if we have a rotation, vectors transform like this between the two coordinate systems for domain 1 and domain 2. And RDC is like this. So we see why it's called a tensor. It's a symmetric 2 tensor. We put the vectors on either side. So RDCs are quadratically dependent on the geometry, but only linearly dependent on the alignment. And RDCs transform like this, and alignment tensors transform like this. So once we have a representation of these things, we can transform back and forth for RDCs, alignment tensors, and vectors in different systems. This is all just linear algebra. In our case here, let's look at what averaging really is. If we had a fixed thing, not average, we would just transform between the two alignment tensors like this, with the rotation corresponding to red to blue. When a biophysicist writes down ensemble average, we have to be a little more careful. Let's say this is an integral over rotation space of the observable times the probability density function. And as it averages back and forth, that's what's actually being fit and recorded here. So our question mathematically is, having decomposed each of these alignment tensors into this nice representation here, using singular value decomposition, rotation, and eigenvalues, can we solve for this function P? That's the question. We're asking for a function here. So how can we do such a thing? So here's the basic takeaway for this. If you write down what you need to transform one alignment tensor into another, you get this thing called a Q matrix, which is a matrix of circular moments whose components are these rotations. This started out by the M matrix from my student Jeff Martin, and Yang Chi, spelled QI, renamed it to Q for some reason. I can't imagine why that was. So this matrix here transforms between these. It's the expectation of all these circular moments here. So this is the sort of framework for doing this. And again, we're trying to compute from the data this continuous Gaussian over rotation space, and we'll have an objective function, which is how well does it minimize the error. So I can't do algorithms here, but let me point you to the paper. Q matrix, by the way, for the mathematicians, is just one of the irreducible but rare representations of rotations. So how do we do this? The lanthanide decouples the global tumbling from the internal motions of the bond vectors in each domain, which is kind of nice. And how do you solve for this distribution? Might be a fairly hairy thing. Well, if we count observables, if we assume that this function is actually Gaussian in a special sense, then it actually turns out with 5 independent alignment media, we could solve this exactly. Closed form exact. Now we can definitely measure that, but when we measure it, it typically isn't all, they aren't all independent. So we typically get less, like 2.5 using Tolman's SVD system. So when we have at least 2 independent alignment, you use a branch and bound algorithm. It's in the paper, but I'm going to sort of hint why that's possible. So it's pretty amazing that you can do this at all. But basically you have intervals for these things, and you have a branch and bound algorithm that gives the distribution that best satisfies the data. Now, what is this assumption that it's Gaussian? I want to tell you what this looks like. So here we have to digress into orientational statistics. What are statistics or distributions over things like rotation space? We chose the Bingham distribution because basically it's on a sphere rather than this complicated space. The sphere is actually the space of unit quaternions, which if you're not using for your rotations, I think you should. It actually is a double cover because each of these is the same as its opposite vector. So this is a nice representation of rotations computationally here, and it has the following parameterization. There's a lot of math here, but let me give you the bottom line, is to represent a Gaussian-like distribution called a Bingham. It's simply an exponential distribution over rotation space. It has 9 degrees of freedom, 3 for the rotation, right, which is an angle and axis, then it has 3 for variances and 3 for covariances. So our model has 9 degrees of freedom, which is shockingly close to the number of observables we can actually measure directly by NMR, so we can fit a low parameter representation. Finally, I want to mention, in the paper, there's all sorts of structure to this. For example, this is the last slide with any hair on it. You can decompose this calculation into an averaging operator where all the averaging happens, and the 2 Q matrices for the 2 isoclinic rotations that comprise what it means to be a Bingham distribution. All the details are in our paper, and that's how we calculate this. Now, finally, when we have the distribution, how could we visualize such a thing? So let me go back and then we'll segue to Terry. Under the hood, it's all unit quaternions, which are the only way you should ever represent rotations in a computer. But we can visualize any rotation by an axis, shown in blue here, and then a rotation about that axis, and vice versa. It's not one-to-one, but it's a good way to represent this. So we can represent rotations in that fashion and get some intuition. And one of our goals here was the following. People won't want to abandon their static models of PDB structures because they're so beautiful to look at. So we wanted to come up with a model to represent a visualization of our PDFs, our probability density functions, that were close to as attractive as protein structures. And this is what we came up with, which again has an axis and an angle and a color-coded distribution. And now I'll turn it over to Terry to tell you how much we like this representation, and we offer it to you for your consideration. And Terry will take over and talk about this some more now. I'll stop sharing my screen. Terry, feel free to take over when you're ready.
Okay, so why don't I go back one slide to what Bruce just showed and try to explain what we're trying to represent here. We call this the disk on sphere representation.
Terry, I don't see your slides for some reason.
Never hit share. Okay, now we should be set, yes? Okay.
Yes.
We call this disk on sphere representation of a 3-dimensional probability density function or probability distribution. There are several things in the literature, several representations in the literature of 3-dimensional probability distributions. None of them satisfied Jane Richardson. When I took them to her and showed them to her, well, Jane, does this convey anything? And she would say no, and sent me back. And finally, we came up with this representation, and she approved it. So it has the Jane Richardson seal of approval. The way to look at it is it is a continuous distribution, but of course, to make this visualization, we had to divide the sphere surface into discrete points just to sample the probability at that particular point. And that point represents the orientation of the Z prime axis of the second domain, the one that is left free to move. Its Z prime axis is what determines the center position of each of these disks. And then on that disk, what we're representing is the probability in a percentile fashion of the orientation of the X prime axis. And so the higher the probability of a particular X prime orientation, the redder the color gets on the disk. As the X prime probability goes down, the color goes down from red down to blue to white. And the advantage of this is that it guides the eye to where the highest probability Z prime orientation is. That's where you see the most red color on the surface of the sphere. And it tells us, it guides the eye to show where the x-axis of the second domain is pointing. And of course, since it's a right-handed Cartesian system, the y-axis is defined by those two orientations. Now, one thing that Bruce didn't mention in his introduction to our interpretation of RDCs is that there's a degeneracy in that observation. It's a characteristic of all RDC measurements. Sometimes the degeneracy is greater than 2, but in our case, it was 2. There were 2 solutions that fit the data nearly as well, what we called solution 1 and solution 2. And I'm showing this in particular to emphasize a couple things. One is you can see that this probability distribution is a long way from what we would call anisotropic, right? Isotropic. It is very anisotropic. So for instance, the back side of these spheres is almost white, meaning that there's definitely a preference for this hemisphere that we're showing at the top in the orientation distribution. And the second thing that's being shown here is our use of an ensemble simulation using some of the same software that the Sachs people use to simulate rotations of backbones, which we did for the 6 residues, the 6 residue linker between the 2 domains. When we did that simulation and generated an ensemble, we found that in that simulation, there were steric interactions that precluded the probability that was shown in solution 1. And on that basis, we chose solution 2 as the real solution and solution 1 as the so-called ghost solution. The other thing that's conveyed here is that we used a kernelized set of probabilities coming from the probability distribution in our ensemble simulation to generate what we call a CDIO, continuous distribution of interdomain orientation. That's what we're depicting in these figures. And so that's important to emphasize here, that if a predictor wants to predict a discrete ensemble of structures, it can be kernelized and turned into a continuous distribution like the one we're showing here. Just to show you the particular utility or functional significance of this, we have determined the structure of the C domain of protein A, which is the second domain in this construct that we're showing. We have determined the structure of C domain bound to the FC subunit of antibodies. By taking that structure and mapping it onto the structure of the 2-domain construct, we can ask the question, how close is the distribution in a simulation? Now, we'd like to do this experimentally at some point in the future, but it has its challenges, its experimental challenges to do this by NMR. But from our simulation, we conclude that the probability distribution of the bound complex is fairly similar to the solution of the unbound 2-domain structure, which suggests to us that whatever has determined this particular CDIO, this particular probability distribution in the sequence of the protein, it is optimized for multi-domain antibody binding. And in fact, we can use that to calculate the thermodynamics of the difference in 2 CDIOs, 2 continuous distributions. the one for the free protein and the one for the bound protein. So just to mention that magnetic alignment with lanthanides decouples the interdomain motion from the global tumbling that all proteins have in solution, and we can use continuous models to minimize the number of parameters. So we're just reviewing what Bruce mentioned here. And the conformation preference that we see, it derives from such a probability distribution. Now, it turns out that because it's a probability distribution and it's a continuous one, we can integrate it. And we can integrate it to determine for two different probability distributions. P1 here would be the free protein probability distribution. P2 would be the probability distribution after binding. And the difference in energy due to the reorientation that must take place in order for the protein to pose this two-domain protein into a conformation that's compatible with antibody binding can be calculated by the difference of those two probability distributions. But because the probability distributions are known, the energy difference is known, and so we can derive from that the internal energy difference of reorientation. Now, this isn't the energy of binding, it's just the energy penalty that the protein has to make to bind. And we can use the famous Boltzmann probability version of the Boltzmann law to calculate the entropy change between the two distributions. And since we have the internal energy and the entropy, we can calculate the free energy difference. And those calculations can be used to anticipate, and this is the numbers that we get from our observed free protein CDIO and our simulated bound protein CDIO. We can calculate each of those energies. But one of the reasons to bring it up in the context here is presumably one of the criteria that we will need in comparing our observed CDIOs to predicted CDIOs is some kind of similarity parameter. And we would propose that this delta U is a reasonable way to compare the similarity. The more similar the two distributions, the predicted and the observed distributions are, the smaller delta U will be. So I think with that, these are the issues that we've discussed. I think Bruce could weigh in here if you want to make any additional comments, Bruce. Go ahead.
Oh, thanks. No, I think the point here is we believe that our representation is a solution to parts of these things. It may not be the solution, but a step in the right direction. The one thing we haven't covered is a example of explicitly counting the number of degrees of freedom in previous models, but I can assure you that for well-respected models, it's often hundreds of thousands of parameters, whereas we're talking about 9 or 18 parameters. So there's an advantage in terms of low-dimensional models. I'd be happy to go into that if there are questions to actually look at a case. But I think maybe in the interest of time, we might just mention that some things that we could talk about is, well, what if it's not a Gaussian? What if there are multiple modes? We have ideas on that. RDCs intrinsically have degeneracies due to the symmetry of the dipolar operator. These are less well characterized for these PDFs. In the future, we also want to incorporate translations through PREs or PRIs or SAXs as well. And of course, this raises a host of interesting questions for design as well as mechanism. Namely, you can imagine in the future that instead of designing to some static structure and then feeding that to your diffusion model, you specified your molecular machine by saying the probability density function you wanted it to achieve and asked, for example, me and Terry to design your linker, or maybe Elaine will be able to do that. So that's sort of the future of this, is not only being able to determine the forward direction of protein structure calculation, which instead of a structure will have a PDF, but going from a PDF to ask what should the sequence of the linker be. Those are the things that we are thinking of for the future. But maybe with that, we should just thank the team and take questions. Very grateful to NIGMS for their support. Yang Chi and Jeff Martin did ton of this work from the experimental and computational sides. Elaine is working very hard right now on her constructs and continuing this work. And finally, it's been a great collaboration with everyone at Duke between our labs. So thank you very much for listening to us, and we're happy to take questions.
Thank you, Terry and Bruce. That was great. I want to just make one point, and then I want to open it for questions. And so just to put a little context for everyone, the idea here would be Terry and Bruce would design a system where they present a challenge to the CASP prediction community. And the challenge would be there are these two domains and attached by a linker, and where the predictors would predict the probability distribution. Well, maybe not the probability distribution, as we said, maybe it's a discrete number of conformations and their populations. And I think we need to define how many conformations, but it would also include their populations. And then those predictions would be taken and assessed against the data, and the assessment against data would use these spherical representations as and the metrics that Terry was describing to assess the quality of the predictions. Terry and Bruce, is that generally right, what I said? That's the structure of the challenge?
Yes.
Yes, and we might be able to present two different systems depending on how far our data gets. One which we've totally worked out, and one which we hope to have worked out within a month. Similar systems, though. I think there's a question also of what do we tell them about the system? Do we tell them this is two relatively rigid domains connected by a flexible linker, or do we just give them the sequence and say this is something that's flexible? What do you get? I think there's a number of ways to structure it depending on how much those are priors, basically. What kind of priors do we want to give the community? And I think either would be reasonable depending on how you want to focus their efforts.
So let's have some questions and then more discussion. I think Shoshona raised her hand. So Shoshona, you want to go ahead?
Yes, very, very interesting. I was just curious, in your free energy difference between the unbound and bound distribution, was this always unfavorable because of the rotational space that was hindered by the presence of the bound antibodies? How did you interpret this?
Yes. In our case, it was unfavorable. I would argue that it will always be unfavorable.
Yes, me too.
Because if the probability distribution perfectly matches the
Yeah.
bound conformation, then the difference you get here for your delta U will be 0, right? It will never be favorable, right? But
it could not be because, I don't know, because you are really, you are binding something really big, which, cannot interpenetrate or something, and has other, other constraints. So
Right.
that's quite important.
Yes, it is. Yes, it is. And it was very important in determining the CDIO that we observed when we
Right.
simulated, right? It was the steric interaction of the antibodies that predominated in that particular calculation. The domains are quite small relative to the antibodies. So, yeah.
Thank you.
Let's see, there's another question here by Susan. Go ahead, Susan.
Yes, really interesting talk. I probably missed this, but I was wondering, you were trying to decide between two possible solutions, and I was wondering if your SACS data was able to give any information, or it couldn't distinguish.
No, we did not use SACS data to distinguish those solutions, and that's primarily because in the polymer physics model we used, we didn't get any interdomain orientational information. That's why we turned to RDCs. Now, it could be, with expertise of someone like you, that perhaps orientational information also can be achieved. If you'd like, I can show you a quick view of the SACS profiles we got and the degree of structure that they have. So this is for the 3, 4, 5, and SPA-N versions of the protein. And this is just using this very simple polymer physics model with really only 3 parameters. The fit to the data was quite good. And so the question is there more data, more information that can be extracted from these profiles? And I do not consider myself a sufficiently expert SACS person to be able to answer that question, but maybe you can from looking at it, Susan.
Yeah, no, what I would recommend is just comparing it to the 2 models and also doing it both in reciprocal and in real space. We've seen that and I myself have a hard time wrapping my head around this, but things that are not apparent in the residuals are coming up in the real space. And perhaps because SACS is a frequency technique. You know, I'm not sure why, but I always do a comparison in reciprocal and in real space for that reason.
Yeah, well, that would make sense. And, you know, it could be that we could build a model using the linker, the CDIO for a 2-domain construct and somehow elaborate models for 3, 4, and 5 and SPI-N in principle, but certainly 3, 4, and 5 Bs, and see what this predicted SACS profile, both in real space and reciprocal space, would look like. I would love to talk to you more about that. Guy has already introduced us via email.
That's right. That's right.
But,
Yes.
and this protein is available immediately for SACS characterization. So it would be great to get going on that. Yeah.
Okay, but I mean, if you've collected it at APS, although you cut it off at 0.25 on your, oh, you did log Q. I'm
Yeah,
not
this
sure
is
how to
log Q.
translate
Yeah,
log
yeah,
Q
log
into.
Q. Yeah.
Okay.
So I would say that probably it would be preferable to do it on the actual construct if we can, on the actual construct, because remember, these did not have that lanthanide binding tag, and yet the other protein does. So to the extent that that changes the story, we probably need to redo the SACS with that construct or something like it. Maybe a 3, 4, 5 version of the LBT'd version of the protein as well.
So if you're doing size exclusion, you can send up to 7 samples in
Okay.
one data collection. So
Yeah, good.
okay. Thanks.
Yes, I want to weigh in on that point. I think that, and I think it's up to Terry and Bruce and Susan whether this is feasible, but I think what would be great is if the same samples that we have RDC data for, and I'm getting the sense this might be 1 or possibly 2, plus 1 which is already published, we would also have SACS data for. Does that make sense? Do you think that's feasible?
I think it does. I want to comment that we didn't, although we collected data on the 2-domain construct, the 2 BDPA B-domain construct, it did not particularly fit the pearl-like necklace model, which is a polymer physics model, because it basically is a dumbbell. And that data, which I have, I could share it with Susan, might in fact have a lot more structural information in it, and that might include the orientational distribution. So that one's definitely worth looking at in more detail, and that's data we already have.
And I want to ask another question, which is, you can imagine some different methods to study the system. One is the RDC-induced orientation of the flexible domain. Another would be SACS. Another is a FRET experiment. So could either Terry or Bruce just talk briefly, what's the kind of advantages or disadvantages of these kind of data for assessing the ensemble or distribution of states?
Bruce, did you want to take that, or I can address the FRET question if that's relevant?
Yeah, maybe you can address the fret question, I think that would probably be good. I think that it does require some modification of the protein, which is always something we have to control for, but I'll let you weigh in on that.
Yeah, so I think Bruce's comments about averaging are particularly relevant with fret, because basically if you were to use, say, a fret pair, say labeled with a fluorophore on each of these domains, you'd basically get one number, the average fret between the two domains. And I think it would be very hard to deduce any kind of orientational information. So you might get a pretty good estimate of the average interdomain distance or interfret
Yeah,
pair.
not rotation. Yeah.
Yeah, but I don't think you would get any kind of rotational
Oh, no.
information.
I
So
should have used the word single molecule fret, which will give you some information of the kind of distribution of those distances.
that's true. That's true. And certainly a distribution of distances. But I don't think you would get very reliable orientational information. Actually, you have to account for the orientational difference in order to interpret the distance. We've considered using DEAR, double electron-electron resonance, as a way to get those distance distributions. But most DEAR experiments are done in the frozen state, and I have a hard time convincing myself that we can freeze our sample and get an accurate depiction of what the sample does in solution. Quenching a structure that is presumably interconverting on the nanosecond time scale seems impossible to do. So that's one of the limitations in getting distance distributions.
One thing we've also considered is PCS, pseudocontact shift information. So if you look at the typical RDC equation, you'll see a dependence in the denominator on the ensemble average of the distance between the two nuclei cubed. And typically we're looking at covalent bond, amide, CH. But with PCS, we have a dependence simultaneously on the orientation and the distance. So in principle, there's a joint distribution being projected onto a one-dimensional distribution in pseudocontact shifts. And we've actually worked out the math for that. There's a 1 over r to the 5th dependence, it turns out, because of the tensor. It's kind of complicated, and we haven't quite got our heads around how to make a reasonable model. But in principle, that's another thing we have on the agenda to try.
One of the interesting things about this project is that it has been driving the theory as we've thought of experiments and collected data for experiments. It's driven the theory development sort of back and forth. It's not like the theory is all set up before we set up the experiment. And likewise, you know, the whole concept of the experiment comes from Bruce's extensive expertise on how to interpret RDCs. So it's a very bidirectional endeavor, and that will continue to be the case. I do think the Holy Grail, and this is what the structure predictors are going to expect from us experimentalists, is the joint distribution for distance distribution and orientational distribution. And it has to be joint, not projected, because you know, clearly there are going to be some orientations that have different interdomain distances than other orientations. So the true depiction of this structural thing is of a flexible protein like this is the joint distance and orientational distribution.
Is it Susan? Susan, do you have your hand up again?
Yeah, I put up my hand again. So I have two comments. One is on the single molecule fret. I've done I collaborate on single molecule fret, and I would say that the biggest value is not in the distance, but in the kinetics of the movement. And so if you don't mind me drawing, just to demonstrate, you could have a fret that looks like this, where it's going back and forth. Oops, it's gone.
Share your screen, share your whiteboard.
I'll do a new whiteboard. And so oops, how do I get to the annotation? I can't figure out the annotation. Huh.
Can you just choose your pencil then?
Will it help if I stop sharing?
I think you already stopped sharing.
Oh, I
Yeah,
did. Okay.
see
Okay.
you.
It only allows sharing with users within the same organ. So let me go ahead and get out of this, leave.
It may be that I have some restrictions because of, concern about hacking. I'm not quite sure. It may be set up that way. But go ahead, finish your point. Maybe you can make the point without the drawing, and then later we'll try to figure out the issue with drawing with these with this Zoom webinar setup we have. Are you there still, Susan?
Yeah, no, I had to come back. It crashed. I'm going to just share screen and then just use the shapes, scribble. And so what I see is there's a difference between this and then something that looks like this, right? It's the same distribution in bulk, but there's a difference in understanding of the kinetics of the movement. And so I see that, I've done a lot of work with flap endonuclease, and even though it's very noisy data, you can really see differences in the kinetics of the movement, and when it's stabilized, when it's not stabilized. We really can, it takes a lot of statistics, but you're really able to see this change in a systematic way, and that's the value of single molecule FRET. Is not the distance per se, but the distribution or the kinetics of that distribution.
Conan, do you want to ask your question, Conan? I'll read it. Conan asks, is there disease indication from certain variants of certain domains, potentially? Yeah, okay. Is there disease indication from certain variants of certain domains, potentially explained by the dynamics of this protein probed and modeled in this presentation?
Yes,
So-
I just wrote back, and Terry can weigh in too. So interesting thing is that SPA-N, as Terry described it, binds a pretty wide variety of targets. And it's thought that its structural plasticity lets it do that. So there two are the Fc region of antibodies and von Willebrand's factor, and there are at least 5 others. So it's thought that this conformational selection from this very, very broad distribution of the 5-domain system enables it to do that. And these are incredibly important. I mean, Staph aureus defends itself or masks itself from the immune system by binding the Fc region of antibodies backwards, so that from a certain point of view in the immune system, it looks like something that's coated with human antibodies. And it doesn't have just one structure with induced fit. It has this very wide distribution that we'd like to understand and how it evolved. And it's probably true that some of the ways that SPA-N has evolved are actually virulence factors enabling it to bind these targets in different ways. And of course, von Willebrand's factor is a clotting factor. It also binds, and there are others. I'll let Terry weigh in this some more, but we believe that there definitely is a biomedical and mechanism connection for how SPA-N does its devious work.
Yeah, I'll just comment. The specific question was, are there mutations that have disease implications? To my knowledge, there are no variants of protein A that have been associated with more or less virulence on the part of the protein in the domain, the N-terminal half that we've been talking about. There has been a a lot of effort trying to make antibodies to protein A, and it's been frustrated by this tendency of protein A to bind antibodies in the wrong direction. But Olaf Schneewind at the University of Chicago has succeeded in eliminating antibody binding, FC binding, that is, by substituting 4 residues with alanine. And we're thinking of using that trick as a way to actually build a sample that doesn't precipitate with this didomain LBT, the lanthanide tagged didomain protein binding to FC or antibodies. The problem is that because FC and antibodies are bivalent and the didomain construct is bivalent, you end up making polymers and you don't have an NMR sample. So we're going to have to knock out binding in one of the 2 domains of either FC or more likely in the protein A. And when we do that, perhaps we'll have a stable sample that we can study.
I try to keep this on time, but I do want to ask one more question, a kind of burning question for me. From the prediction point of view, suppose someone just takes the sequence and drops it into Anton and runs a long MD simulation, microsecond, tens of microseconds, 100 microsecond. Would that be the solution? Or do you know if MD can actually simulate the distributions? What are your thoughts about that?
Go ahead, Bruce.
Well, I think it's pretty, I mean, it's an empirical question. I mean, let's talk about MD is this wonderful technique, but let's talk about what it can do and what it can't. So what do we know about the system? So we know that proteins aren't chaotic, but models of proteins are. So the bad news is for any simulation, we're guaranteed that the MD simulation is going to diverge exponentially over time from the true trajectory. On the positive side, we're guaranteed that in an NVT controlled system that's set up properly and expensively, that if we do enough simulations, and the number is very high, that one trajectory, but we don't know which one, will, if you do enough simulations, be arbitrarily close to the mean trajectory. And that's, of course, the basis for the statistical thermodynamics that you can use for free energy perturbation and MD to estimate binding affinities. That having been said, any particular snapshot is one for which, unless things are fully, equilibrated and very carefully set up, there's relatively little evidence for it. So kernelizing those is quite risky without mapping the energy landscape. And finally, most people don't do NVT control simulations because they're super, super expensive, and also because the kinetic energy, the velocity, tends to increase without bound, without using additional things that then spoil that guarantee. So I would be, reluctant to speculate whether it would really work. I think that there's so many parameters that the MD simulation could be fit to existing data if we had knowledge of it ahead of time, but I don't know if I would trust it for this without some empirical measurements of kinetics that could be used to at least fit the exponential curves we're seeing from the observables we're simulating from the MD. So with enough fitting, yes, but out of the box, I don't know if it would work. But, we can empirically test it. And of course, people that do that kind of work are incredibly sophisticated. And I'm always surprised by the things that they can simulate. So it might be possible.
Yeah. CASP attracts different kinds of prediction methods. And, there were traditional kind of molecular mechanics modeling methods, and which made a lot of progress in homology modeling. And then more recently, a big impact of AI. And then, it's an open question what AI methods might be able to do with intrinsically disordered regions. But in this competition, this activity, we may have we may attract more people from the physics community, which I think is kind of exciting, actually.
Well, I think if they come up with a MD-based ensemble and it can be kernelized, it can be directly compared, and we can see what it does. And maybe it will do great, and maybe it won't. We can find out. I'd be very surprised if it didn't capture some of the sterics that we're seeing and some of the constraints from the linker. Probably just depends on how expensive it is and how how expensive and extensive it is. But it would be interesting to see, and it might encourage people to do that.
I'll just bring up something that was mentioned last month at the same meeting, which is that a key characteristic for successfully predicting at least our CDIOs is that each of the members of the predicted ensemble, whether it comes from an MD trajectory or anywhere else, each member has to have an accurate determination of its population in order to predict the overall probability distributions. And so that, of course, means that you better have a very accurate energy for each one of those states. If you do, then those probabilities will be useful. But the challenge is getting the energies of each individual conformer approximately right.
Yeah, and of course, that's a very forefront area right now in the AI methods, whether you actually get Boltzmann populations, whether you actually learn about energies of the alternative states. So it would be interesting that way. Well, I'll ask, I don't want to hold people too long. Clearly, we have to work on the specification together, what we'll ask the predictors exactly to provide. But before we end, I just ask if anybody else who wanted to ask a question didn't get a chance to, wants to say something. Well, thank you, Terry and Bruce. That was great. I learned a lot. I hope this experiment will work. I think that it'll be valuable to your research program to have the connection with different methods that people may bring to the table here.
Absolutely.
I hope it'll work.
Absolutely. Thank you so much for having us and for the great questions and discussion. We all really enjoyed it.
Thank you very much.
Look forward to it.